Welcome to this lecture on the fundamentals of biomolecules. Today, we're diving into a truly fascinating topic, the specificity of enzyme action. Have you ever wondered how your body manages millions of chemical reactions every single second without everything descending into absolute chaos? The secret lies in the incredible precision of enzymes. In this session, we will explore how enzymes act as highly specialized molecular machines, capable of picking out one exact molecule from a crowded cellular soup. We will understand the physical and chemical rules that make this possible, and we will even learn how to calculate this efficiency mathematically. Let us get started. To begin our journey, let us imagine you have a massive keychain with thousands of different keys, but you need to open your front door. Only one specific key has the exact shape, grooves, and ridges to fit perfectly into your door's lock and turn the mechanism. If you try to force a different key, even one that looks very similar, the door simply will not open. In the bustling, crowded environment of a living cell, enzymes act exactly like these highly specialized locks, while the molecules they work on, called substrates, act like the keys. An enzyme will ignore thousands of different molecules floating around it until it bumps into the one specific molecule it was designed to bind. This demonstrates an incredible ability to pick out just the right partner from a chaotic molecular soup. Now that we have an intuitive understanding, let us look at the formal biochemical definition. Enzyme specificity is defined as the ability of an enzyme to selectively recognize and bind to a particular substrate to catalyze a specific chemical reaction. This selectivity is entirely driven by the unique three-dimensional structure of the enzyme's active site, which is the small pocket where the reaction actually takes place. For a substrate to bind successfully, two conditions must be met. First, it must have geometric complementarity, meaning its physical shape fits snugly into the active site. Second, it must have electronic complementarity, meaning its electrical charges align perfectly with the amino acids lining the enzyme. There are different levels of this specificity, ranging from absolute specificity for a single molecule to group specificity for a family of related molecules. Building on that definition, why does this high level of specificity matter so much in real life? Without it, our cells would descend into absolute chemical chaos, with enzymes randomly destroying or building the wrong molecules at the wrong times. For example, the enzymes that break down toxins in your liver must specifically target harmful chemicals without accidentally destroying the essential proteins your body needs to survive. In the medical field, pharmaceutical drugs are designed to take advantage of this exact concept. Drug molecules are engineered to mimic the specific shape of a natural substrate, allowing them to bind and block a disease-causing enzyme. If enzymes were not specific, a drug meant to cure a headache might accidentally shut down your heart. This highlights why understanding this concept is crucial for everything from basic survival to modern medicine. Before we move on to the mathematics, let us visualize this concept with a diagram. Here, you are looking at a classic representation of the lock and key model. On the left, we have a large blue enzyme with a very specifically shaped pocket, which is the active site. Notice the three different molecules approaching it. The green round molecule and the yellow spiky molecule simply do not have the correct geometry to fit into the pocket. However, the red molecule has the exact complementary shape. On the right side of the diagram, you can see the successful formation of the enzyme substrate complex, where the red molecule fits perfectly into the active site. This visual perfectly captures the essence of geometric complementarity that we just discussed. Now that we have seen how specificity works visually, let us understand how biochemists measure it mathematically. To measure how specific and efficient an enzyme is for a particular substrate, we use a value called the specificity constant. This constant is derived from Michaelis-Menten kinetics. The formula is the specificity constant equals Kcat divided by Km. Let us break down these symbols. The symbol Kcat represents the turnover number, which tells us how many substrate molecules the enzyme can convert into product per second when working at maximum speed. The symbol Km represents the Michaelis constant, which measures the enzyme's affinity for the substrate. A lower Km means the enzyme binds very tightly. By dividing catalytic speed by binding affinity, we get a single number. When Kcat increases, or when Km decreases, the overall specificity constant becomes larger, indicating a highly efficient enzyme. With our formula established, let us apply it to a real-world scenario involving the enzyme hexokinase, which adds phosphate groups to sugars in your body. We want to see how it reacts with D-glucose. We are given that for D-glucose, the turnover number, Kcat, is 200 per second, and the Michaelis constant, Km, is 0.05 millimolar. We choose the specificity constant formula to evaluate this interaction. We substitute our values, dividing 200 by 0.05. This gives us a final answer of 4,000. This large number tells us that hexokinase binds to D-glucose very tightly and processes it very quickly. This value of 4,000 will serve as our baseline for comparison in the next steps. Having calculated the baseline for D-glucose, we now need to prove mathematically which sugar the enzyme actually prefers. To do this, we must evaluate the alternative substrate, D-fructose. For D-fructose, the enzyme has a lower turnover number of 100 per second and a much higher Michaelis constant of 1.5 millimolar. We substitute these new values into our specificity constant formula, dividing 100 by 1.5. The result is approximately 66.67. This much smaller number indicates that the enzyme struggles to bind and process D-fructose. The higher Km means the fructose molecule does not fit well into the active site, and the lower Kcat means the reaction is sluggish even when it does bind. Now we reach the final step of our mathematical proof for substrate preference. We compare the two results we just calculated. The specificity constant for D-glucose is 4,000, while for D-fructose it is only 66.67. By taking the ratio of these two numbers, we find that hexokinase is roughly 60 times more specific for D-glucose than for D-fructose. In a physical sense, this proves that if both sugars are present in your bloodstream, the enzyme will almost exclusively choose to react with D-glucose. This stark contrast highlights how sensitive the enzyme is to even slight changes in the sugar's chemical structure, perfectly demonstrating the concept of enzyme specificity in action during cellular metabolism. We have just proven how enzymes choose between different sugars, which brings us to an even more extreme level of precision, stereospecificity. Enzymes are so incredibly specific that they can actually distinguish between left-handed and right-handed versions of the exact same molecule. Imagine your own hands. They have the same fingers, but you cannot perfectly superimpose your left hand onto your right hand. Molecules can exist in these mirror image forms too. Because the active site of an enzyme is a rigid, three-dimensional pocket, a mirror image molecule simply will not fit. Its chemical groups will physically clash with the walls of the active site, preventing a snug fit. Let us look at a mathematical example to see just how dramatic this physical mismatch can be. To see stereospecificity in action, let us examine an enzyme called L-amino acid oxidase, which is designed to process L-amino acids. We will start by calculating the specificity for its natural substrate, L-alanine. We are given a turnover number of 50 per second and a binding affinity, Km, of 0.1 millimolar. We use our specificity constant formula to evaluate this baseline efficiency. Dividing 50 by 0.1 yields a specificity constant of 500. This shows a strong, healthy interaction between the enzyme and its intended target. The low Km proves that the active site easily recognizes the L orientation of the amino acid, confirming that the enzyme is well optimized for this specific biological task. Having established the baseline for the natural L form, we will now prove mathematically how poorly the enzyme performs when the three-dimensional shape is reversed. We evaluate the mirror image molecule, D-alanine. Because it is the wrong shape, the enzyme cannot bind it properly, resulting in a terrible affinity where Km is 100 millimolar, and a very slow turnover where Kcat is just 0.5 per second. Dividing the extremely low turnover number of 0.5 by the very high Km of 100 gives a specificity constant of just 0.005. This tiny fraction proves that the enzyme is practically useless at processing the wrong stereoisomer due to the physical mismatch in the active site. We conclude our proof by calculating the exact fold difference in specificity between the two mirror images. We take the specificity constant of the L form, which was 500, and divide it by the specificity constant of the D form, which was 0.005. The final calculation shows a staggering ratio of 100,000. This means the enzyme is 100,000 times more specific for L-alanine than for D-alanine. Physically, this tells us that the active site is perfectly molded to fit the L form, while the D form simply cannot fit. This extreme mathematical difference is exactly why our bodies can safely rely on enzymes to never make a mistake when choosing between mirror image molecules. We have covered a lot of ground today, so let us summarize the most important ideas. First, the specificity of enzyme action is the fundamental principle that allows enzymes to select and react with one exact molecule out of thousands, acting just like a lock and key. Second, this incredible precision is driven by the physical shape and chemical properties of the active site, requiring both geometric and electronic complementarity. Third, we learned that we can mathematically measure this preference using the specificity constant, which combines how tightly the enzyme binds and how fast it works. Ultimately, this specificity is the essential biological safeguard that keeps our complex metabolic pathways running smoothly, efficiently, and without chaotic side reactions. You now have a solid understanding of how enzymes achieve their remarkable precision.